And all right, you are set to begin whenever you would like.
Okay. So, hi, I'm Amanda. I'm a PhD candidate in Brazil. And when I was making this presentation, I just realized that the title of my talk is a little bit too focused on my study system. But actually, what I'm going to talk about today is how does multiple mutualistic interactions shape the evolution of mutualistic traits. So when we go to nature or when we see a picture like this, the first thing I can imagine is the interactions that are happening there. For example, seed dispersal or cleaning or pollination. And each of these interactions are mutualistic interactions in which individuals benefit from these interactions because they exchange products or service that are hardly for them to obtain. But nature is a little bit more complex than this, right? So we can see individuals interacting with seed dispersers and rhizobia, anemones interacting with clownfish and zooxanthellae, and also pollinators interacting with plant defense. So partners of multiple mutualisms, they can interact with each other and they can affect the outcome of another mutualistic interactions. So here I'm going to focus on the effect of plant defense and on pollination. So each of these mutualistic interactions, they can increase plant fitness. So when we have pollination, we have fruits and we have seed set. But when we have plant defense, the ants reduce the herbivory. But ants can also repel the pollinators. So I'm going to show you here a video of ants being a little bit annoying against a bee. So this bee is repelled from the flower, and this could have some sort of ecological cost for plants. So this is what I tested in my first chapter of my dissertation. I evaluated the effect of ants on pollination. So here on the x-axis, we have the effects of ants on bees or butterflies, and on the y-axis, the Hedge effect. Basically, this effect means that when this is negative, we have a negative effect of ants on bees or butterflies, so shorter visits, for instance, and we have a positive effect on these floral visitors. So what I found was basically that ants have a negative effect on bees, but not on other flower visitors like butterflies. So if we have this sort of ecological costs, we could also expect some sort of mechanisms preventing the co-occurrence of both plant defense by ants and exclusively bee pollination. And this is what I showed in my second chapter of my dissertation. We showed that all plant lineages involved in pollination, exclusively bee pollination, they usually are not involved in plant defense by ants. However, we still have around 50 plant genera involved in both mutualisms. So we could expect, since these plants are involved in both mutualisms, that they should have some traits that ameliorate these joint costs. So here I'm going to focus on three sets of traits. The first one is related to the access of flowers. So ants will not visit the flowers very frequently. So these plants or plants with extrafloral nectaries could have larger pit cell size or larger sepal areas, prevent ant access to these flowers. The second set of traits could be related to pollinator attraction. So if you have more pollinators, if you have more bees, ants will not be able to repel all of them simultaneously. So for this, we evaluated the flower size and floral displays, thinking that larger flowers and larger floral displays usually attract more pollinators. And finally, we evaluated effects on post-pollination traits, which are related to the likelihood of fecundity. So we measured enteropore, stigma diameter, and hercogamy. Hercogamy is the measure between the anther size and the stigma size. So this is my hypothesis. We hypothesized that Chemicrysta lineages with extrafloral nectaries should invest more in flower traits, preventing access to flowers, or reducing ant effects on pre-pollination or the effects on post-pollination. And to investigate this hypothesis, I went to multiple herbariums around the world, and I measured a bunch of flower traits. So I measured petal size and sepal area for traits preventing and access to flowers. I measured flower diameter and petal area as a measure of flower size, enterpart area, the stigma diameter, gynoecium length, and stamen length. For floral display, I counted the number of flowers and I divided by the number of leaves available. And finally, for heterogamy, I measured the size of the gynoecium and I divided by the longest stamen just to check this distance. And I measured all these floral traits in 600 specimens from 164 species. And currently in the phylogeny, we have 231 species. So I measured around 70% of the phylogeny. Okay, so what did I found so far? So all my figures, they will be very similar. Okay, so in the x-axis, we'll have the flower diameter. And I used the flower diameter in my analysis because of these traits could be involved in some allometric relationship with the flower size. In the y-axis, I'll have the trait, in this case, the petal length. Each dot will represent the mean value for each species, gray dots when EFNs are absent, green dots when the species have extrafloral nectaries. Okay, so let's my results. So for pet cell size, we found no difference in the pet cell size for plants with and without extrafloral nectaries. For sepal area, again, no difference. So this means that this species with extrafloral nectaries don't have any floral traits preventing access to flowers. For my second hypothesis, in which I measured flower size and floral display, we found no difference for flower size, so no difference for petal area, and no difference for flower density. So basically, again, Chemicristis species that have exophoral nectaries don't have floral modifications that reduce the end effects on pre-pollination. So they are not more attractive to pollinators in this case. For my third hypothesis, so I measured the anther-pore area and I found no relationship for between plants with and without exophoral nectaries. Again, and plants with and without exophoral nectaries have no difference in the stigma diameter. And for heterogamy, so unsurprisingly, no difference again. So plant species with exophoral nectaries don't differ in traits in relationship to post-pollination effects. So basically, I rejected my first hypothesis, my second hypothesis, and my third hypothesis. And after panicking, thinking about all of this, what we showed is that basically this conflict between ants and bees have not been a major selective force driving the evolution of floral traits in Chemicrysta. But okay, so what? Why? Why? What explained our results? We were thinking about some mechanisms. The first one's that this plant species, this plant lineage is pollinated in a system called bus pollination. In this bus pollination system, the bees grabs the anther and they vibrate their thorax, and the thorax are in contact with the anthers. So by vibrating the anthers, they release the pollen. So it's a very specialized system. So maybe some slightly changing in floral traits could have a huge implication for the pollination efficiency. Also, all Chemicrysis species, they are all bus pollinated. But extrafloral nectaries, conversely, they are highly labile and they might have evolved in plant lineages only when the benefits of herbivory reduction offset the pollination cost. So here we have Fabaceae clade. The color dots represent some sort of extrafloral nectary types. And in white, we have the loss, or again, of the loss actually of the extrafloral nectaries. So we have multiple losses of extrafloral nectaries in this phylogeny. But yeah, okay, we measured all morphological traits, but it's possible that this plant lineage, they have other mechanisms. For example, they might have some sort of chemical repellents. So when it goes to visit the flower, they are repelled by these floral repellents. And floral repellents are really common in plant species involved in obligatory mechanisms, in obligatory mutualisms with ants. So they might be common in other types of mutualisms, like this one mediated by extrafloral nectaries. Another mechanisms could be the temporal segregation or phenological decoupling between the extrafloral nectaries secretion and the pollination. So extrafloral nectaries, they extrafloral nectar, it's usually secreted when plants are investing new leaves. So maybe when plants are young, while pollination could happen when plants are a little bit older. So there may be some sort of temporal segregation between these two mutualisms, allowing the coexistence between them. Okay, so I showed you that we rejected all my hypothesis, but my results are still cool. At least I think it is still cool. And mainly because many studies evaluating these multiple mutualistic effects, they focused on the ecological costs at the ecological scale. And here we measured these ecological costs at the evolutionary scale. And we showed that the morphological traits is not involved in this reduction in costs, but other traits could be other mechanisms could be involved in this reduction in these costs. Okay, so as take-home messages from this presentation, I would like you to keep that the NTB conflict do not affect the evolution of floral morphological traits, at least in this plant lineage, and the evolution of extrafloral nectaries might have relied on alternative strategies, for example, chemical repellents or temporal segregation, and that the effect of multiple mutualisms might not drive the evolution of traits beyond the pairwise interaction. And with that, I'm happy to answer any questions you have. And here's my contact if you want to send any email. And that's it. Thank you for listening.
All right, and we've got plenty of time for questions. So you can either use the raise your hand feature if you want to ask it verbally, or you can go to the Q&A feature if you would prefer to type it out. So open the floor for some questions. All right, so Catherine, let's have you unmute.
I was just curious. I liked all these. Thank you for your talk. And you had a lot of really interesting ideas from some other traits that might be involved in mitigating some of these costs. Have you collected any data on these other traits? Just a sneak peek on future things. Just curious.
Unfortunately, I don't. I would love to have, but I don't. We don't have the phenological material for many of these plant species. We are trying to work on the chemical repellents right now, but it's gonna take a while for do that. But yeah, we don't. Not for now. Maybe in the future.
So we also have a question that has shown up in the Q&A. So the first one there, we have somebody asking that they were curious about the idea that the ants disturbing bee pollination has a negative impact on the plants. And is there any evidence suggesting that pollination by butterflies is more advantageous or disadvantageous than pollination by bees?
Yeah, no, we don't have any evidence. What we think is happening is because when bees are pollinating, they are in close contact with the flower, while butterflies, they can be a little bit away from the flower. So it's probably because of the morphological coupling between the pollinator and the flower, but we don't have any evidence that the pollination by butterflies could be more advantageous or disadvantageous. But maybe being pollinated by more animals could be advantages for this plant species.
All right, so we probably don't have another time for another verbal question. There is one more question in the Q&A, but we can also have our next speaker start sharing their screen. So Meaghan, if you'd like to. And then we'll have you wait, the additional 25 seconds until we get to the start of your talk.
Excellent. Thanks, Mike.
All right, you are free to begin whenever you wish.
All right, thank you. Hi, everyone. Thanks so much for being here today. It's such an honor, and I'm so excited to have the opportunity to present here in the Hamilton Symposium. And today I'll be sharing a story from my dissertation about inbreeding and land use change in populations of a threatened rattlesnake. So from Gregor Mendel's experiments with pea plants to Darwin's fascination with plant and animal breeding, evolutionary biologists have long been interested in both how inbreeding occurs and its impacts on phenotypes, individuals, and populations. Studying inbreeding is becoming increasingly critical in the modern world because of wide-scale human land use changes. As an example, from 1850 to 1920, cropland in the conterminous United States expanded by 1.15 million square kilometers, which is about the size of California and Texas combined. And although timescales vary, many of these changes in land use have occurred rapidly over the past few hundred years, giving wild populations little time to adapt to these new conditions. And the populations that remain in these habitat fragments are often small and isolated, leading to concerns about increases in inbreeding, loss of genetic diversity due to drift, and inbreeding depression, and ultimately concerns about the ability of these populations to persist. And so some really critical questions to think about when studying inbreeding in the Anthropocene is, does habitat fragmentation actually drive individual inbreeding in some of these fragmented populations, as well as testing for fitness effects of inbreeding called inbreeding depression? And here I'm going to address these questions using what I think is a really fascinating and perfect study system for studying inbreeding and habitat loss, the Eastern Massasauga rattlesnake. Eastern Massasaugas are found throughout the Great Lakes region of the United States and Canada, and they have some pretty specific habitat requirements. They need open wetlands with adjacent woodlands to overwinter. Because of the kind of natural patchy distribution of these types of habitats, they have likely existed in fairly small populations throughout history. However, they've also experienced a lot of habitat loss over the past few hundred years. These plots here show what this landscape in the United States looked like using some backcast land use and land cover data in the years 1630 compared to 2020. And we can see that a lot of natural areas have been converted to agricultural and urban land use types. This is a problem for Eastern Massasaugas because they do not like these sorts of land cover. Anthropogenic land cover is a significant predictor of population extirpation. Largely in part because of this habitat loss, Eastern Massasaugas are currently listed as threatened in the United States and are a species of special concern in Canada. Over half their historic populations have been or are expected to become extirpated. And so we know that this habitat loss drives population extirpation, but what we don't know is how habitat fragmentation impacts inbreeding in these small and isolated populations, and if inbreeding has any fitness consequences in this species that was likely historically small and patchy. And so today I'm going to tell you about how we're seeking to understand patterns of inbreeding both across the entire range of Eastern Massasaugas and if they're correlated with severity of habitat loss, as well as focus in on a few populations in particular where we've been able to actually test for fitness consequences of inbreeding. To address questions about inbreeding across the range of Massasaugas, we have whole genome sequencing data from 26 populations. We have around 2 million high-quality SNPs with an average of 19x depth of coverage. And we also drew on backcast land use data from Lee et al. To look at variation in habitat loss in the area surrounding these same populations. So this plot here on the x-axis is the proportion of natural land use type, and the y-axis is going back through time. And we can see that around 200 years ago, the areas around these populations started losing natural habitat types. But this has varied between populations, with some experiencing a lot of loss and some remaining relatively consistent. To look at inbreeding across the range of Eastern Massasaugas, we used runs of homozygosity. A run of homozygosity is a long stretch of homozygous genotypes inherited from both of an individual's parents. We can calculate the proportion of an individual's genome that's found within a run of homozygosity, and this is called F-rho, and it's a measurement of individual inbreeding. And we found from our whole genome dataset that inbreeding is generally high across the range of Eastern Massasaugas, with some individuals having as much as 50% of their genome being found in a run of homozygosity, which is a lot of homozygosity. And we also found that this varied between sites and between different regions. For example, now I've these boxes of color in the background represent kind of different geographic regions. And we can see that snakes from the range edges in like Ohio and Pennsylvania have higher inbreeding compared to northern Michigan, where we generally see lower levels of inbreeding. So because we see this variation in inbreeding across populations, we can then ask if the history of land cover change predicts this variation. So to summarize the often messy and complicated temporal history of land use change over the past few hundred years, I isolated a 2 by 2 kilometer area around each sampling site and calculated the average proportion of anthropogenic land use from each year from 1630 to 2020. So a value closer to 1 indicates widespread and long-term disturbance, and a value closer to 0 indicates relatively little disturbance. I then built a series of candidate regression models, some including the average proportion of anthropogenic land use and some without, and used AIC comparison to select the best fit model. Our best fit model did include an effective disturbance history on inbreeding. So more disturbed sites typically have higher F-rho than less disturbed sites. However, you've probably noticed that there are many individuals with high F-rho or high inbreeding that are from sites with little to no disturbance. And this likely reflects that there are many other factors that explain F-rho, things like long-term demographic history and population bottlenecks. As an example, some individuals in this low disturbance category are actually from a population on an island that's been very isolated and likely experienced a lot of genetic drift over time. So using this whole genome data set, we find evidence of elevated inbreeding across the range of Eastern Massasaugas, though typically higher at range edges. And we find that some variation in inbreeding is explained by habitat loss and disturbance. So even though populations of Massasaugas are naturally patchy and small, they're still being impacted by human mediated habitat loss and fragmentation. So next, I tested for inbreeding depression in 2 populations of Eastern Massasaugas and investigate possible factors that promote inbreeding. So these 2 focal populations are located in southwestern Michigan, and we chose these sites because they are the sites of 2 really amazing long-term monitoring projects led by my wonderful collaborators, Jen Moore, Eric Heilman, and Lisa Faust. Because of their dedication over the past really 15 years, we have capture-recapture data from over 1,000 unique individuals across both sites. And luckily for me, at each capture event, blood is taken from snakes, so I was able to extract DNA from all individuals and genotype them at approximately 2,000 neutral loci spaced evenly throughout the genome. I used the SNP dataset to calculate FGRM, which is an estimate of individual inbreeding suitable for this low-density SNP dataset. And I also used this genetic data to reconstruct empirical pedigrees for each population to measure reproductive output. I did this by just counting the number of individuals assigned to a genotyped parent. Then I used zero-inflated negative binomial models and AIC model comparison to select the model with the best fit to the data. This model did include an effect of inbreeding, or FGRM, on the number of pedigree offspring an individual was assigned. However, the effect size of this relationship is rather small, and the confidence intervals overlapped with 0, so it's hard to say if this is biologically relevant. What is relevant is that we see a significant relationship between inbreeding and the probability that an individual had any offspring, regardless of number. A 0.05 increase in FGRM is associated with a 53% decrease in the odds of having any offspring in the pedigree. So When inbred snakes reproduce, they only have slightly fewer offspring than non-inbred snakes, but they are much less likely to reproduce overall compared to non-inbred snakes. Because we're doing all these comparisons within a population that shares a demographic history, I was curious what might be generating this inbreeding we're seeing within populations. And what I found was that there's very fine-scale spatial genetic structure within these sites. This was somewhat surprising because these sites represent pretty small geographic areas. The maximum spatial extent is under 2 kilometers. These plots here show centroid spatial locations of individuals from capture events. Each dot is colored by that individual's score in a principal component analysis. And you can see that the dots are roughly clustered together by color, indicating that more genetically similar individuals are found closer together. We tested this result statistically using the pedigree data and found that pairs of related individuals like mothers and offspring, fathers and offspring, and pairs of parents are all found significantly closer together in geographic space compared to pairs of unrelated individuals. And we think it's likely that the life history of eastern Massasaugas is generating these spatial patterns. First of all, eastern Massasaugas have extraordinarily limited dispersal. The maximum distance between capture locations of any snake over the course of 10 years of capture-recapture data was 960 meters. So they're not moving around very much. Additionally, males will look for mates in the same geographic locations every year. And there are aspects of maternal care that suggest that young of the year might actually follow their mothers or other related females to find critical overwintering sites that are really important for their survival. And so they're hypothetically all these really interesting aspects of Massasauga life history that are impacting their genetic patterns. And so here we present what we think is the first direct evidence of inbreeding depression in rattlesnakes. And we tie this to kind of some key life history elements of the species. So in conclusion, by using genomic data, we found that across the range of eastern Massasaugas, habitat loss and land use changes do explain some variation in the elevated amounts of inbreeding we see in these populations. Critically, we think this is pretty important for managing and conserving these endangered and threatened populations. I didn't mention this before, but these 2 populations highlighted by blue boxes, these were actually some of the last snakes we ever found at these sites. And we think that they've become extirpated. Obviously, this is anecdotal, and we can't tie their extirpation to these elevated levels of inbreeding, but it does suggest a concerning trend. By using long-term monitoring data as well as genetic data, we were able to test for inbreeding depression and found that inbred snakes are reproducing less often compared to non-inbred snakes. And I think this emphasizes that some species may just be at higher risk of inbreeding depression than others due to their life history traits. And so it's important to take kind of a holistic view of demographic history, contemporary habitat fragmentation, and kind of basic biology when helping populations persist in human-modified landscapes. So with that, I'd like to thank my collaborators and funding sources, as well as you all for listening.
All right, so we have time for 1 or 2 questions. We do have one thing already in the Q&A asking if there have been other relevant changes in land use at the sample sites beyond the natural to anthropogenic.
Yes, that's a great question. There have been. So Massasaugas are also pretty reliant on kind of an open canopy. They want like these really sunny areas where they can bask. And so as wetlands kind of naturally go through succession and become more closed in, that also makes them kind of unsuitable habitat for Massasaugas. So a lot of invasive species that kind of shade in these wetlands are an additional problem. So one future direction we're excited to take this work is to try to take kind of a more holistic view of what a Massasauga would actually consider suitable habitat rather than just using this kind of binary.
Okay, we do have one other question. I'm gonna have to direct you to put that into the chat because we've got like 30 seconds until our next speaker begins.
Excellent.
Sorry
Thank
about
you.
that, Nick. All right, so our next speaker, if you would like to share your screen, Kip. Then you can begin whenever you are ready.
Awesome. Can everyone hear me? Okay.
We can hear you. Hopefully you can advance your slides.
I think I can.
Okay.
Thank you to the organizers for having me, and thank you for all listening to me. I'm going to tell you today a story about a parthenogenetic ant, and I'm going to do so starting by reminding us all how sex and meiosis works. So I'll start in the germline of a female chicken who is diploid and who each of her cells contain two copies of each homologous chromosome. So these are largely identical, except for a few DNA base pair differences between the homologs, reflecting this homozygosity down the length of each chromosome. And as she undergoes meiosis, this will start with DNA replication, followed by crossover recombination. Homologous chromosomes will be pulled apart in meiosis 1. Sister chromatids pulled apart in meiosis 2. The chromosomes will segregate randomly through these meiotic divisions according to Mendel's law of segregation. And this will produce 4 haploid nuclei. In female meiosis, one of these is selected as the egg, and after an analogous process occurs in the male germline, sexual fertilization will produce a diploid offspring that undergoes many rounds of mitosis, and the process continues. So that's how the birds do it. It's also how the bees do it, and it's basically how the vast majority of animals reproduce. But of course, not all animals reproduce this way because asexual reproduction evolves sporadically. And when it does, this occurs via perturbations of sex and meiosis. So incipient asexual lineages sort of face two proximate challenges. First, they need to restore ploidy without sexual fertilization, but they need to do so in such a way that maintains viable genotypes. And that's important because the ways of restoring ploidy have different effects on the genotypes of offspring. Maintaining viable genotypes often means maintaining heterozygosity, both to avoid recessive lethality, to kind of avoid the kind of inbreeding depression that Meaghan was just telling us about, and then also to maintain genotypes viable with female development, compatible with female development. And although we know about a large number of asexual taxa, we rarely know how they meet these two challenges. But by doing so, we can hope to learn new things about how meiosis and sex work, how they evolve, and therefore how genes can flow through populations. So with that in mind, I've been studying the clonal raider ant, Ossuaria biroi, which undergoes basically a cytologically normal meiosis, followed by what we call a central fusion. And I'll show you what that looks like cytologically. These are pairs of homologous chromosomes that align during metaphase, are pulled apart during meiosis 1. Sister chromatids are pulled apart during meiosis 2. The scale bar has shifted, and now these are 4 haploid pronuclei, each of which contains 14 homologous chromosomes. So for an insect female meiosis, this is completely cytologically normal so far. And if it were to continue to be so, then this innermost nucleus would migrate over, fuse with the male pronucleus, and then that would become the diploid offspring, and then these other pronuclei would degenerate. But what happens in O. biroi is that the central nuclei migrate together and fuse. Those become the diploid offspring, the others degenerate. And to me, this has always seemed like a really elegant way of restoring ploidy without sex. But if you look to the cartoon on the right, you can see that this can incur loss of heterozygosity if you inherit one recombined and one non-recombined chromosome after a crossover. If you just draw out all of the possible inheritance outcomes, there's 4, and you expect them with equal probability because of Mendelian or random chromosome segregation. 2 of those lead to loss of heterozygosity, and 2 of them don't, meaning that for any one crossover, you should expect 50% of the time to lose heterozygosity. And that's always seemed to me like a really steep cost. So when I set out to investigate this system, my first question was, exactly how much heterozygosity do we expect to lose? And for that, I turned to binomial models, where I'll just take as my trials the number of chromosomes in the haploid karyotype. I assume 1 crossover per chromosome, which is canonically the minimum, basically, and then a 50% probability of losing heterozygosity. And that just tells us very simply that we would expect on average 7 chromosomes to inherit these large chunks of loss of heterozygosity. And then we would expect in 99% or more of meioses for at least 1 chromosome to have a big chunk of loss of heterozygosity. And then only very rarely should there be no losses of heterozygosity. To see what that looked like empirically, I sequenced whole genomes. Here's a histogram along the genome of a mother. And what you're seeing is the number of SNPs, single nucleotide base pairs per window, variations per window, that are heterozygous in that individual. And what I expected to see was that in her daughter, there would be several segments of chromosomes that had lost heterozygosity. But that's not what I saw. So here are the real data. And here I'm just showing every site at which there was a difference in the maternal and daughter genotype. And here, this was just a single SNP that lost heterozygosity. In a second mother-daughter pair, the same story, except there was just a single SNP that then gained heterozygosity. And so this is really not what we were expecting. There's none of these big segmental losses of heterozygosity. So why might this be? The only reason we expected losses of heterozygosity is because we assumed that crossing over occurred during meiotic prophase. But if there was actually no crossing over in meiotic prophase, then it wouldn't matter which way the chromosomes segregated. As long as you do a central fusion, you'll never lose heterozygosity. So to see whether this might be the case, I flattened oocytes at metaphase of meiosis, and I looked for physical crossover structures, or chiasmata. And here I'm showing with red arrows paired homologous chromosomes that seem to be connected by putative crossover structures, these chiasmata. But I really wanted to have genetic evidence for crossing over. And for that, I turned to a quirk of ant biology, and I'll sort of explain that here. I told you before that most of these meioses are followed by a central fusion, which produces diploid offspring, and these develop as females. But occasionally, in this species, meioses are not followed by fusion, and that produces haploid offspring that develop as males. That's really rare, but males and females are sexually dimorphic, so you can almost tell males from females by eye, which is useful, and I'll show you why. These are 2 hypothetical genotypes that differ from each other by 3 crossovers. If you sequence the whole genome of these with short-read sequencing, you get the genotype at every locus. And so if you actually try to detect these crossovers, 2 of them you can't because the genotype is actually identical. But for this third crossover, now you can detect this because there was a loss of heterozygosity distal to that crossover. But then if you take those same hypothetical haplotypes and put them into hypothetical haploid males and hypothetically sequence their genomes, you can now detect all 3 of those crossovers. So the punchline here is that crossovers that occur without loss of heterozygosity can be detected with haplotypic data, but not with genotypic data. These haploid males occur rarely, so I sort of opportunistically sampled them. They're not from known pedigrees. There's presumably tens or hundreds of generations since their last common ancestor. And so I sampled diploid females from the same sort of scheme, sequenced their whole genomes, and performed all pairwise comparisons to count crossovers. And what I found was hundreds of crossovers among haploid genomes, but fewer than 10 crossovers among diploid genomes. The same story in 2 different clonal backgrounds, just emphasizing that it really does seem like, rather than crossovers being absent from meiosis, crossovers seem to occur, but without loss of heterozygosity. So I draw on here mother, daughter, granddaughter, but I actually at this point didn't know the timescale. And to do that, I would require pedigree-based studies. So I would need haplotypic data from diploid individuals. And so for that, I turned to linked read sequencing, where in contrast with short read sequencing, you can then phase haplotypes and then directly compare those haplotypes to count crossovers. I'll just show you again the 4 different inheritance outcomes following a crossover. You could either inherit one, but not both recombined chromatids. You could inherit both recombined chromatids, or you could inherit neither. And I can only detect a crossover by sequencing, say, mother-daughter pairs if at least one of the recombined chromatids was inherited. And I just say this because in total in this experiment, I detected 144 crossovers by sequencing multiple different pedigrees. And under Mendelian expectations, we would expect that 96 of those should have been associated with a loss of heterozygosity. But in fact, only one of them was. So this blew me away. It's very strongly and statistically significantly non-Mendelian, and it really begs the question of what exactly is happening here. And there's really only two possibilities. One of them is that actually all four of these possible genotypes are produced, but then losses of heterozygosity are lethal, and so then I only really sample individuals that didn't lose heterozygosity. I'll just go ahead and say that's a little bit unlikely because it kind of requires recessive lethals on multiple chromosomes and a lot of assumptions. But the other possibility is that rather than segregating randomly through the meiotic divisions, there's non-random segregation such that you always co-inherit the reciprocally recombined chromatids. To get a sense of the relative roles of each of these in maintaining heterozygosity, I turned back to my binomial models. Again, just showing the number of chromosomes with a loss of heterozygosity each meiosis. And I can substitute in my empirical number of crossovers, which was a little lower, and that already sort of increases the probability slightly of inheriting 0 losses of heterozygosity. But to modulate this more, I wanted to vary the probability of co-segregation between completely Mendelian levels and completely non-Mendelian levels. And that can allow us to really jack up the proportion of offspring that don't inherit a loss of heterozygosity. And then further, I could subtract a proportion of individuals that died due to losing heterozygosity, which allowed me to model the proportion of offspring that inherit loss of heterozygosity, with green values being high proportions and yellow values being low proportions. In total, in this study, I sequenced 15 offspring whole genomes, and none of them inherited a loss of heterozygosity on any of their chromosomes, which, if you do the 95% confidence interval, allows you to say that basically the only realistic parameter values fall above this black line, meaning that you either need very high homozygous lethality or very high non-Mendelian co-segregation or some combination. And then further bounding this, homozygous lethality is hard to measure, but what you can do is just pretend that all mortality results from a loss of heterozygosity, and you say that's the upper bound. And so I basically measured mortality rates during development in replicate colonies. And then I just say that basically that's the upper bound of the effect that homozygous lethality could have. And so from this, the realistic parameter values that could explain our data are way up in the upper left corner in this blue box, meaning that really homozygous lethality plays at most a minor role in maintaining heterozygosity and possibly none. And then there really needs to be strongly non-Mendelian co-segregation of recombined products to maintain heterozygosity in the system, an 800% increase over the Mendelian expectation. So this is cool for a number of reasons. One of them is that there's other cases of documented non-Mendelian segregation, but in all those cases, there's particular alleles that bias their own transmission, and so it's sort of selfish, sort of like these selfish centromeres that are more likely to end up in the egg than into the polar body. But this is really unselfish because no allele has biased transmission. All alleles are biased to be co-inherited. So the what's cool is that even though this is a pretty unique scenario, there's similar phenomena that have been identified in an asexual nematode and also in human oogenesis. In the nematode, it's also associated with maintenance of heterozygosity. And in human oogenesis, it's that chromatids with a recombination event are more likely to be inherited than ones without. And just looking out, if we really want to understand what's going on, that'll take more detailed molecular and cytological studies. But really, this unselfish meiotic drive could actually be more widespread than we currently appreciate. It's difficult to detect, and the only reason I detected it is because I was studying a system using phase genetic techniques that inherits 2 different products of female meiosis. And those other 2 studies are some of the only other ones that have comparable studies in animals. So if we really extend this technology and use phase genetic studies of multiple products of female meiosis in diverse animals, including sexual species, we might find that this is really another lever by which evolution can tune the rate at which recombination events get inherited and therefore modulate the way that alleles flow through populations. So with that, I'll thank you guys for listening, and my advisor, Daniel Cronauer, and other folks, and I'd be happy to take any questions.
Just to let you know, the questions will need to be in the Q&A because you're like right out of time now, actually. Need you to stop sharing so we can get our next person to share their screen.
Great.
All right, so our next speaker, can we get you to share your screen,
Yeah.
Gina? Yep.
Yeah, let's see. Okay.
All right, and you are good to go right now.
Okay, awesome. Let's see. Okay, within our DNA strand are promoters and gene bodies, and within those are CPG islands, which are long stretches of the DNA that contain high concentrations of C's and G's. When methyl groups are attached to the cytosines, we call that DNA methylation. So within our promoter regions, the cytosines are typically unmethylated, and in gene bodies, these are typically methylated. Now, in that way, epigenetics alters the gene expression without altering or changing that underlying DNA sequence. In experimental systems, when mammals are exposed to a stressor, not only are the animals directly exposed, but so are their germ cells. In females, intergenerational inheritance occurs down to that F2 generation and down to that F1 in males. And then methylation marks that are passed down transgenerationally are past that. However, in observation-based systems, any transmission of methylation marks are called multi-generational transmissions, and they can essentially be thought of like an epigenetic memory that is passed over time. It's possible that these multi-generational transmission of epigenetic markers provides avenues for adaptation to new and stressful environmental conditions by preparing those future generations to deal with those stressors. However, it's important to acknowledge that epigenetic heritability is not in itself evidence of adaptation. These epigenetic marks may be neutral with respect to a phenotype and fitness, or simply they might represent a stressed or a disease state. Therefore, with that in mind, we were primarily focused on two main objectives. The first was to quantify the maternal and the paternal influences on the epigenome over several generations, and that'll allow us to understand the drivers of adaptation that occur due to methylation. And then after that, I'll discuss its influence on fitness. So typically these research questions are difficult to study in a wild system. However, with such a data-rich system like the banner-tail kangaroo rats, it was possible. So here we have 4 subpopulations that are geographically restricted to a very small area with strong population fluctuations and density-dependent behaviors. We also utilized a 17-generation pedigree to evaluate the genetic relatedness of these individuals. We sampled individuals so that they, their parents or grandparents, and great-grandparents were sequenced such that we could take 1, 2, and 3 generation comparisons of our methylation markers. So we divided our reference genome into 11,500,000 base pair windows with 250 base pair overlap and filtered out windows with high missingness and those with no variation because they were indicative of highly conserved regions. And we calculated the methylation percentage per window, and that resulted in 14,784 windows for downstream analysis. We used pairwise correlations of parents and offspring divided by sex across all 14,000 methylation windows with the 1, 2, and 3 generation comparisons to determine the multi-generational patterns of heritability. And we considered those windows as significantly heritable when they were greater than 1 standard deviation from the mean. So here we have the number of generations removed on the x-axis and the number of heritable windows on the y-axis. And we're seeing that there's that drop-off of epigenetic heritability over time. And with that maternal line, that is somewhat consistent throughout time, whereas that paternal line is driving a lot of that influence in that first generation, but it drops to only 1 significant window in that 3rd generation. And that's really exciting because it conforms to the theory of epigenetic heritability in mammals that we know from lab-based systems. And that's when we know that sperm-contributed methylation marks are transmitted, but they're not stably maintained beyond that F1 generation. And of course, this is likely due to shared environmental pressures during gestation or due to maternal parental care early on in life. But then we wanted a more detailed breakdown of the multi-generational heritability. And here we have density curves. So you can think of like those bell curves on the y-axis or on the x-axis, and the correlations on the y-axis. So a higher correlation is depicted higher on these figures. And on the left side of the figure is that paternal line, and on the right side of each of these figures is that maternal line. So in that first generation, we're seeing that both the maternal and the paternal are really influencing those high correlations, but that paternal line is driving most of those higher correlations. By that second generation, the paternal line is decreasing in those correlations, while that maternal line is driving a lot of those differences. And then by that third generation, we're seeing essentially a normal bell-shaped curve centered around a correlation of 0 in that paternal line, with that maternal line driving a lot of those differences. So that allows us to depict the multi-generational structure of epigenetic investment in offspring in a wild population. What's cool about this is that the asymmetric decay could be adaptive, and this is especially true in unpredictable but spatially structured habitats like we have in the banner-tailed kangaroo rat system. We also know that there are density-dependent dispersal in which small population sizes are resulting in further dispersal across these subpopulations. But what's really cool about this is that females are twice as likely to disperse these long distances compared to males. So what we're seeing in this epigenetic profile is that these paternal methylation inputs show these short-term environmental match, likely due to that close proximity to that home mound, with a fast decay over time, where that maternal methylation input is having those long-term environmental memory with that slower decay over time. But then how does that relate to siblings? Because we have the 17-generation pedigree, we were able to find full and half siblings of the same and different sexes, all from the same mom, all born within the same year, so that we could determine the heritability of methylation marks across siblings. So across all 14,000 windows, we calculated the absolute differences in methylation percentages and considered those as significantly heritable when they were less than 0.7% of a difference in the methylation profile. Here we found that full siblings had 26% more heritable windows than half siblings of the same sex, and over 3 times as more heritable windows than half siblings of different sexes. And also half siblings of the same sex had almost 2.5 times as many heritable windows as half siblings of different sexes. So we're seeing not only the influence of genetic relatedness on the epigenome profile, but also in sex. So that's telling us that that parental investment of methylation marks in siblings have similar proportions genome-wide, but at different windows throughout. And we know that genetic variation is important for population persistence, so it has been suggested that epigenome diversity can act as a buffer for populations with low genetic diversity, like we have in this system. This could also be a form of parental bet hedging, in which the mother will give different portions of the epigenome to their offspring in hopes that 1 or 2 of them have strong fitness to be passed down to that future generation. So, with that, we were able to quantify the maternal and the paternal influences in a wild system. And then now I want to shift to discuss how we examine the influence of methylation on fitness. And that's allowing us to know more about the evolutionary insights into the genetic drivers of a population's stability. So now that we had those heritable methylation marks, we also wanted to characterize methylation marks that are regulated by the environment. So we grabbed windows with the lowest 0.5% of variation. We wanted to be super conservative here, which resulted in 888 environmentally regulated windows across our 12 years, with 865 of them unique, which means that most of those windows that were represented more than once weren't in subsequent years. And that then told us that the environment fluctuated sufficiently across these years to influence variable methylation, even across these short distances. So after some initial data exploration and sifting through the substantial metadata that we had, we conducted ridge regressions to examine the influence of methylation on fitness. So using the significantly heritable and the environmentally regulated windows and some significant metadata like the mound's location and the month of sampling for our predictors, we then use longevity, or an individual's lifespan, and the lifetime reproductive success, or the number of offspring that reach maturity, as response variables for our fitness measures. So here on the x-axis, we have the different models with the different response variables. And on the y-axis, we have the number of significant windows. We're seeing that there are 70 significant windows that influence longevity and about 125 that are influencing lifetime reproductive success. So that's telling us that there are various environmental and natural history predictors that influence how heritable and environmental methylation marks will influence fitness. But when you look at this, you might be wondering, why are there about half as many significant predictors for longevity than there are for reproductive success? And we're seeing that these longer timescales likely requires more methylation marks. So when comparing the timescale for measures of fitness and their effects on inbreeding level, longer timescales require more markers. So here in this figure, these red numbers are the estimated number of lethal equivalents, which can be thought of like the relative number of genes that must be deleterious to influence mortality. And the number of lethal equivalents for survival are approximately a third of those for lifetime reproductive success, which is really similar to the patterns we're seeing here in the epigenome. And with that, I find it super exciting because once we have the context of these methylation patterns across even more taxa, we can better target our expectations of evolutionary processes based on how we know that the epigenome influences things like adaptive capacity and fitness. So with that, I'd like to thank the many people that had a hand in this work, especially Dr. Averill Harder, who put together our reference genome, and Dr. Peter Wasser, who and his students, who collected all of these data and made it possible to do this type of work. And I'd like to open communication lines so that we can discuss this work in the future or answer any questions that you may have. So thank you for listening.
All right, so we probably have time for a couple of questions. We've got a couple already in the Q&A feature. I don't know if anyone wanted to jump in with something verbal, but your first question in the Q&A is asking, where was the population studied?
Yes, this population is in the desert in Arizona. Yeah.
Okay, and then our next person is asking that they noticed that you used ear punch samples for this, and isn't DNA methylation variable across tissue, so why did you choose this particular tissue type?
Yeah, good catch. Yes, ear function, yeah, so the epigenome does vary across tissues. However, ear tissue is very important, or an ear function is very important for the species, for their evading predators and for interspecific communication. So they require low, low frequency sensitivity in their ears. And so this body part is super important for their survival. And then it allows us to ask these questions about fitness by using this tissue.
So you do have a couple of other questions. We're not going to have time to do them verbally right now because we're going to have to move on to our next speaker, but you're free to answer them in there. And then we also only have 5 speakers in the session, so we will have some time for additional questions at the end. Jeremy, if you would like to share your screen.
There we go. My screen should be shared.
Yes, it is.
Excellent. All right.
So wait about 30 seconds. We're going to keep things on the actual timer.
All right. Excellent.
All right, and you are free to begin.
All right, thank you. So first, I would like to thank the organizers of the Hamilton Award for selecting me and letting me share my research here. I'm Jeremy Summers, a graduate student at Nancy Chen's lab at the University of Rochester, and today I'm going to share my work for my thesis that I'll be defending next week that investigated the demographic and fitness consequences of increasing isolation in a natural population. There we go. So to set the stage, populations around the globe are becoming more isolated due to habitat loss and fragmentation. And this isolation can have immediate demographic effects that threaten these populations, either due to direct decline in immigration or changes in demographic structure of that population, such as in the age of individuals. In some cases, like this example of the woodchuck shrike, previously stable populations are revealed to be sinks that can go extinct once immigration is cut off. There's also long-term genetic effects of isolation. Fewer new alleles are entering via gene flow, which means that genetic diversity will be lost, which can impact the ability of the population to adapt to future change. Additionally, fewer unrelated immigrants can lead to increased levels of inbreeding. This can lead to inbreeding depression, the decline in fitness of more inbred individuals, which can add up to even affect entire populations, like in this case of resident orca whales, where steadily increasing levels of inbreeding resulted in population decline. However, the effects of immigration can change over time as the population becomes isolated. Density dependence and interactions with immigration and other demographic rates can buffer the population against the effects of isolation, resulting in demographic compensation. Gene flow that still does occur can also reduce levels of inbreeding, reversing inbreeding depression and resulting in genetic rescue, such as with the Florida panther. After new individuals were introduced, the previously inbred population rebounded due to increased outbreeding. Yet the fitness effects of gene flow can change over generations as well. In a study on an island population of song sparrows, Dickel and colleagues compared the fitness of new immigrants and their descendants to residents, shown here with the posterior distribution of those comparisons. Male F1s who have one immigrant and one resident parent had much greater lifetime reproductive success than their mean of each of those parental groups. This fitness benefit, which is known as heterosis, where the greater heterozygosity of those outbred individuals increase their fitness, largely due to the masking of deleterious recessive alleles. However, male F2s, the offspring of those two F1s, had significantly lower fitness than their parents. Instead of benefiting from being outbred, F2 suffered a fitness cost due to the mixture of immigrant and resident alleles, known as outbreeding depression. This shift from fitness benefits to fitness costs can play a major role in the effectiveness of gene flow. Few studies have investigated how the effects of change of immigration over time can impact populations that are becoming isolated, despite the fact that these populations exist at a crucial time point when early action can prevent future crisis. Part of the reason for this gap in knowledge is just the data required to study immigration over time. We need to identify resident and immigrants. We need to collect detailed demographic information on the population over a timescale where immigration changes, and ideally we need multi-generational fitness data. Thankfully, there's a study system that meets these requirements. The Florida scrub jay is a federally threatened bird restricted to fragmented habitat patches across its historic range. These birds have limited dispersal, they're non-migratory, they remain on well-defined territories year-round within family groups, and they're also cooperative breeders, where young delay dispersal for multiple years and remain on their natal territory, assisting with rearing siblings. This limited dispersal has made the Florida scrub jay ideal for long-term monitoring. A population at Archibald Biological Station has been individually tracked for more than 50 years, with complete breeding records and records of all immigrants that enter the population. Together, this dataset has allowed us to construct a population-wide pedigree up to 16 generations deep that is also molecularly verified. This is perfect for studying the multi-generational effects of immigration. This population is also useful because it is becoming isolated due to the decline of smaller surrounding populations. Immigration rates into Archibald were historically high, but have dropped dramatically over the past 30 years. During this time, immigration rates have remained pretty much low. Thus, the population at Archibald behaves like a sink that is being cut off from its input. Due to the drop in immigration, there's been an increase in levels of inbreeding, with the mean breeder inbreeding coefficient rapidly rising once breeder turnover caught up with the population. What makes this picture more complex is that the immigrants that enter Archibald are themselves less heterozygous than residents, and thus likely more inbred. While immigrants may help with outbreeding, they may also harbor novel deleterious genetic variation that is maintained in their source populations due to genetic drift. Given this population on well-maintained habitat facing isolation, I wanted to investigate the demographic and genetic consequences of immigration. I wanted to know whether isolation itself poses a threat to this population, and if there are any demographic compensation that's occurring. I also wanted to see if gene flow had any long-term effects by investigating the fitness effects of immigrant ancestry, and if these benefits persist beyond the first generation of outbreeding. We can start by looking at the population growth rate over the past 30 years, shown with this black line. The growth rate fluctuates around 1, which is stability, meaning the population alternates between growth and decline. However, the geometric mean of the growth rate, shown with green, is right at 1, so the population has remained demographically stable overall. To determine the source of this stability, I used a path analysis to link temporal variation to the population growth rate. The variation that I included had food availability, population density, climatic cycles, the increasing level of inbreeding, and any other independent trends, such as the decline in immigration, captured by a time term. I then used generalized linear models to link these factors to underlying demographic rates that define the Photoscrubjae life cycle, such as survival, fecundity, and immigration. And then finally, I used a matrix population model to link those demographic rates to the population growth rate, allowing us to link all the way from sources of variation to growth and decline. Looking at the results of those generalized linear models, we found that juveniles and male breeders that were more inbred had lower survival, and more closely related experienced breeding pairs had lower fecundity, matching previous results that found inbreeding depression within our population. I also found density dependence in both male breeder survival and female immigration, indicating that density dependence could play a role in stabilizing the population as immigration declined. And finally, immigration did decline over time, as expected. However, breeder survival also unexpectedly increased. Could this trend in breeder survival be compensating for the effects of isolation? We directly tested for compensation by simulating the population over the past 30 years and controlling which temporal trends occurred. The base simulations, with all trends, closely matched the observed results, with deviations caused by outlier years. We then used a female and male-specific model due to slight differences in demographic rates between sexes. Once we removed the increasing trend of breeder survival, the population declines. However, if we also remove the decrease in immigration and the increase in inbreeding, the population then becomes stable once again. The restoration of stability means that breeder survival is specifically compensating for the consequence of isolation. Looking back at our initial questions, we did find evidence that isolation poses a threat, but compensation by breeder survival prevented decline. These results paint the population as shifting from a sink that relies on immigration to a more independent but isolated population. However, immigrants and residents are not identical, so we need to consider the long-term fitness effects of gene flow as well. The first step to do this is to compare the fitness of residents and immigrants, which reveals the additive genetic effects of immigrant ancestry. Assuming an additive genetic model, the offspring of residents and immigrants, F1s, will have the mean fitness of their parental groups. As mentioned earlier, the increase in heterozygosity of F1s can lead to heterosis due to dominance effects. These effects will present as deviations from the expectation of F1s when using an additive model. F2s, the offspring of two F1s, have the same amount of immigrant ancestry as their parents, and thus are expected to have the same fitness under an additive model. However, recombination allows F2s to be homozygous for immigrant or resident alleles at different loci along the genome. This reduces the effects of heterosis and introduces epistatic effects, where loci interact. These epistatic effects are what caused the outbreeding depression in the song sparrow population previously mentioned. Finally, F1s can pair with residents or immigrants to create resident and immigrant backcrosses, who have the same genetic effects as F2s, but will have their fitness shifted towards their non-F1 parent. Across each category, we compared the realized fitness of individuals to their expected fitness based on the effects already accounted for by their parents. This reveals the underpinnings of any fitness effects of immigrant ancestry. We categorized all individuals in our study tract based on the number of immigrant grandparents they have. By using grandparents, we focused primarily on recent immigration, which has changed over the study period. Also important to note, this means that some individuals that are classified as immigrants were born on our study tract, but have entire immigrant ancestry. We then used a Bayesian generalized linear mixed effect model to estimate lifetime reproductive success for breeders, controlling for whether an individual dispersed into the study tract, their cohort year, and the identity of their parents. The results of our models are then presented like this, with the posterior distributions of lifetime reproductive success relative to residents. For example, the 95 credible interval for immigrant and immigrant backcrossed females do not overlap 0, indicating that both groups have much greater lifetime reproductive success than residents. This increase in fitness for immigrants indicates that there are positive additive effects of immigrant ancestry in our population, though primarily in females. These additive effects likely explain why all females with some immigrant ancestry tend to outperform residents. Next, we can compare these distributions to the expectations that we have based on that additive model. The realized fitness of male F1s is then greater than the expected value across 85% of its posterior distribution, which provides support for dominance effects, specifically heterosis. Similarly, immigrant backcross females have greater fitness than expected across 89% of the posterior distribution, supporting either effects of heterosis, not apparent in F1 females, positive epistatic effects, or possibly a parental effect, such as if having an F1 or immigrant parent itself improves lifetime reproductive success. These results support that despite high historical immigration and reduced genetic diversity of immigrants, there are fitness benefits of immigrant ancestry that persist for multiple generations. Altogether, the demographic and genetic consequences of isolation paint a complex picture in this system. The well-preserved habitat at Archbold likely enabled this population to shift from being maintained via immigration to adjusting to isolation, which is good news that focusing on preserving habitat can overcome the demographic effects of fragmentation. However, even at an early stage in isolation, recent immigrant ancestry increased individual fitness, meaning the efforts to maintain gene flow would already have immediate benefits. Yet these benefits are sex-specific, which should be considered when planning programs such as translocations in scrub jays or in other systems as well, as it does seem to be showing up whenever looking at multi-generational fitness. These outcomes then lead to future questions to understand how populations persist through isolation. The demographic shift in our population was accomplished via increased breeder survival. This outcome could be a direct response to immigration, or may be caused by other co-occurring trends, including the introgression of previous immigrant alleles. The benefit of immigrant ancestry that we found may also depend on the source population of immigrants. These populations may be more or less diverged from residents and can harbor different levels of genetic load. Understanding how variation among immigrants by source population impacts the fitness effects of gene flow can help us create stable metapopulations and ensure assisted gene flow efforts are effective. And with that, I would like to thank all my funding sources, as well as everyone in the Chen Lab and at Archbold that has made this project possible. And I would be happy to take any questions at this point, or if you want to reach out on any of my contacts presented here, please do so.
And we do have plenty of time for any questions. So as always, either raise your hand or put something into the Q&A and we can handle it that way. We just had something show up in the Q&A. So Andrea says, cool work. I may have misunderstood, but it seems like you were saying that epistasis does not play a role in F1s and aren't classic DMIs just epistatic effects in F1s.
So within F1s here, the epistatic effects aren't going to be as present with, or it's going to be requiring dominant effects there. Because it's not going to be interactions necessarily between loci because you can't be homozygous for immigrant or resident alleles. You can have epistatic effects that are over-dominant or are dominant covering up the recessive abilities. So if you have two incompatibilities that can have an effect even when they're heterozygous, those would be present in F1s.
And then Catherine, go ahead.
Yeah, great talk. Thank you. I saw that you looked at overall ancestry. Just curious, do you see particular regions of the genome with immigrant ancestry that are conferring these fitness benefits in the immigrant females?
So right now we have just completed a massive effort to do whole genome sequencing across more than 3,800 individuals in our population to be able to investigate this. We are actively now working on trying to identify any of those regions that could be associated with this benefit of immigrant ancestry. This is also how we're going to be looking at trying to assign immigrants to their source populations as well. So this is a really exciting direction that we want to head in.
All right, well, that was our second round of the Hamiltons. There are still other talks going on in other sessions. If you wanted to pop over to one of those, we do also have this time in case anyone wanted to ask any other questions of our speakers. Otherwise, you might take advantage of having a slightly longer break until our next set of sessions, which are going to begin up in an hour and 15 minutes from now.
Thanks, everybody.
So I'm going to change the controls such that anyone is able to unmute themselves